My name is Jan. I am a postdoc at the University of Groningen, and I work with Bert Pullman and Matthias Heinemann. And here in Groningen, we are not working with the GCVI minimal cell, but rather we are interested in creating a synthetic metabolism for the synthetic cell of the basic and now evolved programs, with the objective of course, producing ATP for the synthetic cell, but also keeping homeostasis. So for those who are not familiar with our work, we work with the arginine breakdown pathway. We call it AGI pathway. This is a very simple route for the creation of ATP inside the vesicles, and it relies on a transporter and only 3 cytosolic enzymes. The transporter ARCD exchanges external arginine for internal ornithine, and then the internal metabolism replenishes this ornithine so that the exchange can keep going. And then it forms carbonyl phosphate, which can bind together with AGP to form ATP. So we have a very simple, with 4 enzymes to form ATP inside the cells. We have reconstituted the system inside vesicles, and we managed to measure ATP formation. And to measure this, we use Perceval. So Perceval is an artificial probe that binds to magnesium ATP and also free ATP and emits light in the form of fluorescence. And then we can measure the 500 and the 430 signals. And then by taking the ratio between these 2 signals, we have a proxy for ATP to AGP ratios. This way we can measure the AGP to AGP ratio inside the vesicles in time. Now, in this talk, I wish to talk about the 2 questions that are very important for the creation of a synthetic cell. So how much ATP we needed to produce inside the vesicles, and then how fast we needed to produce this ATP. So the first question has been answered in this 2019 paper by Hendrik and Bert and others, in which they considered major cellular processes, and then by summing the ATP requirements of each individual process, they arrived at the number of 3.6 times 10 to the 8 ATP equivalent to sustain a cell cycle of a synthetic cell equivalent in size to the GCVI. So one question that one may ask is if you could, for instance, produce all this ATP beforehand and store it inside the cytosol and then use it for the cell cycle. Well, if you calculate the concentration of ATP with all of these ATP equivalents, we see that the concentration would be 20 molar, which means, of course, that we cannot store the ATP. We have to produce it on demand as the cell cycle progresses. Now, this triggers the second question. If you needed to produce it on demand, then we have to match the requirements of the cell cycle at that specific moment. So then we needed to know how fast this ATP needs to be produced by our synthetic metabolism. And we also needed to know if our current system meets these expectations of ATP production. So let's then dive into it. This is how our experiment, typical experiment looks like. Here on the vertical axis, you are seeing the ratio of Perceval, which is basically ATP to AGP ratio. We have a calibration curve to convert between them. And then you see that when we add arginine to the external of the vesicles, we have a phase number 1 in which ATP is produced, and then we have a second phase in which ATP is converted back into AGP, and then we finally have an equilibrium. So in this system, we have only ATP production inside the vesicles, but we don't have anything to consume the ATP so far. That's why the ATP accumulates. But our next step is to add something to consume the ATP and then, for instance, produce the lipids for the vesicles to grow. So that when we do that, we are going to be in the phase number 1, in the phase of ATP production, which is, by the way, the fastest ATP production phase. So if we now zoom into phase number 1, we have this figure over here that we can use to estimate that the ATP in our system is being produced at a rate of 0.08 millimolars of ATP per minute. Now, using this rate, we would take 19 days to produce all these ATP equivalents in order for the cell to double in volume. So of course that the ATP is being produced too slow, which means that our system is under optimized. That's where my work came in. So what we did here to tackle this problem was to develop a mathematical model of the age pathway. And in this slide, I will quickly explain why we need a mathematical model for it. So let's say that we wanted to optimize the age pathway. So this is a very simple pathway, so it should be easy to do that. Well, it turns out that this is actually very difficult to do. So for instance, if we wanted to optimize the ATP production, we could, for instance, make the uptake of arginine faster. And to do that, we could, for instance, increase the substrates for ARCT, for instance, ornithine. However, if we do so, we may end up with more arginine inside the vesicles. And then the next enzyme, which is ARC-A, we discovered that ARC-A is inhibited by arginine. So if you have more arginine, eventually the ARC-A will become slower, which may make the ATP production slower. So this is a very non-linear problem. We have a lot of trade-offs. This is one of them. The other one would be that by having more ornithine, the next reaction by ARC-B, which moves in the thermodynamically unfavorable direction, would be very compromised. So we really needed to remove the ornithine from the vesicles in order to produce ATP. So I hope I have convinced you that even this very simple four-enzyme system is already very complicated in behavior, so that we need some kind of mathematical model in order to understand its dynamics. Now, the way we built this model here in Groningen was to separate the four enzymes and then kinetically characterize each of them in solution. So we did that for the transporter, we did that for ARC-A, B, and C. I'm not going to dive into the details, but here in the figures, I am showing you the characterization of ARC-B as an example. We used HPLC to measure the formation of product, while we variated the substrates. This figure over here is in the thermodynamically favorable direction, and this one in the unfavorable direction. And then the characterization is like we are fitting in, for instance, a Michaelis-Menten equation. But here, of course, our enzymes are more complicated, so we have to go for more complex models, such as the ordered by mechanism. You are seeing the equation here. And then by fitting this model, the model are the lines here on the graphs. By fitting the model, we obtain all the kinetic parameters that determine the dynamics of ARC-B, for instance. After doing that, we also considered some relevant side reactions. For instance, we have a buffer inside the vesicles, and then we dynamically consider the buffer species within the vesicles. We also consider other protonations that are relevant for our model. And then we finally consider the passive membrane diffusion of byproducts, such as ammonia and CO2, which are produced inside the vesicles, but they are toxic and they need to leave, and they leave by passive diffusion. So all of this is considered, and then after characterizing all of these reactions, we can assemble the model, which is basically a bunch of differential equations, as you can see here, master equations, as we call them in physics, in which on the left we have the variation of a given metabolite inside or outside the vesicles, which is related to the velocity of the reactions that we have characterized previously. In these slides, of course, I am summarizing years of work, but then after that, we managed to get this model, which is the black lines on the graphs, and then the color lines are our data measured with Perceval. So you can see that the model describes very well all of the data. And then after obtaining this model, we now can use it to optimize the ATP production, which is our main objective. So to do that, here I have an example of how we can do that. This is an example in which I allow the ARC-D, which is a transporter, amount to variate in the membranes, and also the ARC-C concentration. This is our standard condition that we usually use in the lab, so 0.08 formation of ATP. In this graph, blue means less ATP production and red means more. And then we can write a program. I use Python to search for the maximum ATP production, give these ranges. So you can see that only by varying ArcD and ArcC concentrations, we already almost double the ATP production. This would be very difficult to get only qualitatively. That's why the model helps a lot to find this maximum. We cannot test all the conditions in the lab. This is just impossible. And then the model helps narrowing down the possibilities to be tested on the lab. This is just a 2D example, but then I also performed the 4 enzymes. Now it's the same thing as before, but now for all the enzymes. And then by doing so, we pass from the standard to the optimized conditions you see here in this table. And then I gave these optimized conditions to my lab mate, Elwin, who performed the experiments. And then you can compare the standard formation of ATP in blue to the optimized formation of ATP in orange, which corresponds to 4 times more ATP production, which then reduces the time to produce all the ATP equivalents back to 5 days, which is way better than, of course, 19 days as before. But as for the next steps, some preliminary simulations already show that we can achieve a 1-day cell cycle. This would be our milestone for now. We can do that by varying also the metabolite concentrations, so they now enter into the optimization. This is yet to be done, but it seems that this is achievable with the system that we have. And then after that, we of course wanted to include the other modules, because now we have only the ATP production, but we have to couple this with other modules, for instance, lipid synthesis for membrane expansion. So this is then the next step. This is going to be modeled as well as we did here. And with this, I would like to thank you for your attention, and then take any questions that you might have. Thank you.
So we could add, I think, 1 gene to our minimal cell system and have, well, no, we could add 1 gene to wild type Mycoplasma mycoides and make the entire this arginine metabolism system work, or we could add all 4 genes to it. Would this be a useful model for your lab?
Yes, this is something that is running in the lab right now. So far, we don't have genes, so it's just in vitro experiments. But then we are already working on this. For instance, we work on optimizing the pathway via evolution, for instance, then in vivo. And then for us, for the model, yes, there are techniques for optimizing when you actually have the genome. So this, yes, is totally possible to include in the model, even though we don't do this right now. But yeah, this is something that we are already working on.
So, but for instance, we could put these 4 genes into our minimal cell system and send you a cell, if that would be something that you and Bert would be interested in.
Yeah, absolutely. So far, perhaps our model cannot tackle everything, cannot have all of this, but yeah, it's totally possible to take a look into this, and yes, we would be interested for sure.
So let me know.
Yeah, sure.
Questions? Zan, I see. No, I see.
Yeah, just a comment. You might know, because of John and Kim Wise, and during our calculations, they asked us to keep keep measuring how much ATP is used in the SYN3A, and we have that. And I always thought, oh my God, these high values, can they possibly be right? But it uses and also produces a lot, because that's the only way it gets ATP. So yeah, I think that's an interesting comparison number. And we have a plot in the Cell 2022 paper. And we didn't put it in the most recent paper, but it might be in the SI. I'll have to ask what my coworker, colleague, Zane Thornburg.
Yeah, absolutely. The numbers are so high. That's why we needed the recycling. We cannot have all the AGP from the beginning. So we really needed this recycling cycle, to make it work.
Yeah. So SYN3A also brings in a lot of magnesium, but there is one gene that's still there in Syn3A that is called FOB, which I believe blocks, it can inhibit the magnesium input. So if you want to try those experiments, John, you might want to also remove FOB, but you do get a lot of magnesium, more than you need for the number of ribosomes and nucleic acids that you produce.
Kim?
Yeah, Jan, really interesting work. A couple of questions about the transporter ARC-D. I understand it's an antiporter. Is that, if that's correct? What kind of energy does it need to run, if any? And the other thing is, if you have a a propagating system, does it require any energy, like Zan is asking, to get ARC-D inserted into the membrane?
Yes, so ARC-D is in fact an antiporter, and it operates based on the electrochemical gradients of the substrate. Yeah. So when we start the system, we have arginine outside, and we have no arginine inside, and then we already have ornithine inside from the beginning. So those create an electrochemical gradient, which then defines all the energy available to run the system. And as the times go, we dissipate this energy and into the formation of ATP. Yeah, but all the energy of the system comes from the electrochemical gradients. But I'm not sure if I understood your second question.
I think you answered everything I was asking, so maybe that did it.
Okay.
Okay.